Respected Director CPPS, External Examiner Dr. Mani Challappan, uh, retired Professor Dr. Agraman, professors, and my dear students. Good morning to each and every one of you. In our university, Dean SPGS has made it mandatory to present a seminar by external examiner whenever they conduct any examination. In this regard, we have actually requested Dr. Mani Challappan to deliver a lecture, special guest lecture on some aspect. He readily accepted and he is going to deliver a lecture on endosymbionts. On behalf of the department, I welcome sir for this seminar, and I welcome Director CPPS and professors and other students for this seminar. Once again, I welcome you all. Now, I requested our Director CPPS to give her introductory remarks.
Good morning to everyone. And in continuation of our person head, I also welcome everyone for this guest lecture. And first of all, I should thank our guest speaker, Dr. Mani Challappan sir. He is my immediate senior during my undergraduate studies at Madurai Agriculture College. And we had interactions during the visit to Velaini when I was the external examiner to that campus. And he is now the handling two different posts. One as Dean Agriculture and also he is the professor head of entomology at Velaini. And he's the specialist in insect physiology and also vertebrate pest management. He has developed number of technologies for the vertebrate pest managers. I am very much interested to hear about this, sir. So after this, we shall have some discussion about this because we are in need of some technologies for the vertebrate pest management, especially the wild boar and also peacock birds. So there are menace in this case of maize for and also different crops. The farmers are facing problems. I have seen your biodata. You're having developed a number of list of technologies for the management of this. And he has done his undergraduate at Madurai and postgraduate in the Coimbatore campus and also PhD in IRA Coimbatore. And he's also serving as chief editor in Journal of Tropical Agriculture in KAU, and also he's worked as agriculture research scientist from 1994 to 99. And after that, he is serving as head person head here, and also as dean. And he is having many award recognition and also awards. And he has selected as Agriculture Research Service in 91 and also passed National Eligibility Test and IRA Senior Research Fellowship, ICR Junior Research Fellowship, and awarded Best Innovative Showcasing of Eco-Friendly Technologies in South Indian Agri-Fiesta. And also he has bagged Best Teacher Award in KAU in 2020 and is also the Fellow of Entomological Society of India. And he has received Best Research Award too in 2021. And he is also serving as external examiner in several SAUs and also in Australia for the external member in Western Sydney University. And also he has guided 6 PhD students as major advisor and 25 as committee member. With regard to PG, he has guided 12 students as advisor and 43 students as committee member, and also guided 4 internship students and handled 19 research projects funded by ICR, RKVY, NABARD, and GOK, and Director of Agriculture, Department of Agriculture, GOK. And he has handled number of extension activities, handled different trainings, exhibitions, and developed electronic and social media to disseminate the eco-friendly technologies. And he has authored 2 books of international nature and also 7 technical bulletins and 11 book chapters and 2 review articles and 62 number of research papers were published by him. And he has attended international training at South Africa during 2017 at International Barcode of Life, and also in 2016 Indo-Australian workshop on stingless bee pollinators was attended at Sydney. And also he has attended Indo-Australian workshop on honeybees and pollinators in New Delhi in 2016, and international training program on small mammals in Kerala in 2009. And different technologies he has involved in generation of these technologies like classical biocontrol of papaya mealybug, nesting design of damer bee, bio bird repellent, P-foul fence, T-net, automatic cracker station, reflective ribbon cutter, rotary avian blind, bird nesting designs, LED light bird scarer, deep prep, and nylon net based wild boar fence, boar rep, solar electric multi fencing system, and also rodent cake applicator, sensor based wild boar sonic scatter, holographic reflective ribbons, and born owl as biocontrol agent in Lakshadweep Islands were introduced by him. So there are a number of technologies, as I told you earlier, a number of technology is developed for the management of vertebrates. So let us hear something addition to this endosymbionts sir, about this also. So with this a small introduction, I invite you for giving us lecture, sir. Thank you.
To respected Director CPPS, the head of the department, my senior colleagues, the students, thank you very much. Thank you very much for giving me the opportunity. I'm the alumni of this campus, this department. So basically, I worked on the insect physiology for my uh, PhD. And later I moved to ICER, and I was an IPM person over there. I was the only one in Goa Institute. Later, I moved to the SAU, where I made an area where many of you can also venture. So that is the topic that I would be presenting today. So this beautiful picture, it's the lush green view. Can you see a living entity over there? You can see a tree and a meadow, right? Is there anything else? Is there anything else? There are hundreds and millions of life over there. But when the earth developed, it was not like this. It was hellish. Everywhere it was a volcano and inhospitable the situation. This is the situation where the magma was flowing, nothing was there, and it was inhospitable. The life started, if you see, the earliest fossils, the carbon, the presence of carbon around 4 billion years. That was the earliest record we have, the presence of life. And 3.7 billion years ago, the archaea was there. And subsequently, the bacteria came into existence. At that time, the oxygenated atmosphere was not there. Around 2.2 billion years ago, the cyanobacteria came into the existence, and it changed the environment from non-oxygenated to the oxygenated. And from there onwards, the multicellular organisms developed, and presently we are at the topmost. We are the youngest in the evolutionary history of life in this world. I just wanted to tell you that the microbes, they are the first colonizers of this world, and they have undergone all odds, and they overcame the inhospitable nature of the earth. I can very safely tell that all organisms are the super organisms of microorganisms. Do you agree with me? You are the super organism of all microorganisms. Do you agree with me? Yes, it is. So during the period of evolution, the eukaryotic cell acquired the microorganism. That is the organism you would be seeing in every cell of your body. The mitochondria, it is having a different genome. It's not yours. The chloroplast, it is totally different. It is not plants. If the chloroplast is not there, the plants cannot survive. Apart from that, there are numerous microorganisms that would be supporting you. If you are happy, it is because of the microorganism, and if you are not unhappy, it is because of the microorganism. The mood of any organism, it is because of the microorganism. The general fitness of any organism is microorganism. So the microorganism is present everywhere, and we know less than 1% of the microorganism that is culturable, because the Sanira sheet used some agar plates to grow the microorganism. We cannot grow the microorganism present in the environment. They are called environmental DNA, and the study is called the metagenomics. Metagenomics, first it appeared in 1998, though the work was started in 1985. And within the span of 2022 years, a lot of paper, lot of literature, lot of studies have come. And we started the work in 2011. When I proposed the work on metagenomics for one of my students, my head of the department asked, why you are doing like this? What you will get out of it? It has a number of applications. And these microorganisms, they are ubiquitous and they are indispensable. Yeah, the students would be knowing what is ubiquitous and what is indispensable, right? Without that, nothing will. I am going to the topic that I have chosen. It is endosymbiont, the hidden players in insects. I just shorten it as an insect symbionts. The symbiosis you would be knowing. It was coined by D. Barry way back in 1879. It is a dissimilar organisms coexist. That is the symbiosis. Whether it can be a mutualism or parasitism or commensalism, and you would be knowing what are all these things. So in the commensalism, the organism would be on the body of the host without giving anything, or the host would not be deriving anything from that. The only the symbiont or the organism which would be deriving the benefit from that. Whereas the parasitism, you would be knowing the both the, the organism too would not be, it is in the negative, you see. Yeah, it is not deriving anything, whereas the mutualism, both would be benefited. So all these things would be considered as a the symbiosis. If you see the microbial diversity in insects, the proteobacteria, it is the largest group that we would be seeing. How to identify? Two methods. One is the culture method. So only less than 1% of all the microorganism you can identify based on this. It is very, very difficult. It's a cumbersome. And the next method is the NGS, next generation sequencing. So here the environmental sample can be taken. From air you can trap, or from the swab you can take anything, any material can be taken, and the DNA can be isolated. Sanira, you used how many insects to get the DNA? Okay, okay. So with the minimum sample, we can get the DNA. And many times when we go for the endosymbiont study and all, we take only one insect, a white fly or a mealybug. We take and we get the DNA out of it. So it is very tricky, very tricky. And this next generation sequencing machine, it gives the MB data, so a lot of data it gives within a short period, so that we can have the complete sequence of that organism. Later, we can go for this type of graph, MGRAST. It gives a beautiful picture, and it gives the relation of all these microorganisms present in that particular sample. So based on the microorganism, we give the functional diversity, how the microorganism would be benefited to that particular superorganism. Just to classify the bacteria, it is divided into three major categories: the proteobacteria, actinobacteria, and tenoricutes. In this particular group, the Wolbachia, it is a universal bacteria, like it would be present in almost all insects, and this is being used as one of the candidate microorganism for controlling the insects also. IIT, incompatible insect technique, it is being used in the mosquito and all. Later I will show you how it is being done. So Wolbachia is a very important candidate that many of us can do research on that. There are a number of other bacteria also, the beta protobacteria and gamma protobacteria also there. And where they live on insects, it is not truly endosymbiont. It would be present on the insect, in many places. On the cuticle, it is present in the gut region without embedding in the cells. It is present. It is present intracellularly, or it would be present in specific mycetocytes that you would be knowing. It is both primary and secondary. The primary endosymbiont, it is very essential, very essential. Without that, the organism will not survive. Whereas the secondary symbiont, the organism will survive without the symbiont also. But it gives some conditional benefits to the host. So these are some of the primary symbionts present in insect. And this microorganism should be transmitted. It should go on in generation together. Mostly the primary symbiont, it will go through the female germ line. So it will be ovarially transmitted. It will be embedded in the egg, or it will be smeared, or it will be in the capsule form. So it is both having the vertical transmission and horizontal transmission. The vertical transmission, definitely it has to pass from one generation to another generation, whereas the horizontal transmission, it can occur from the environment also. That is the difference. So this is how it is transmitted, the transovarial transmission that you would be knowing. So the symbiont, it would get into the egg and it goes the outside, during oviposition. Whereas the capsule transmission, along with the egg, it also goes as a separate entity. And egg smearing, we know the larva comes out, the lepidopterans or colopterans, they eat away the egg shell, right? So they would be occurring these microorganisms. So horizontal transmission, when the egg is laid, it is asymbiotic, and later on it occurs. And this there is a mechanism that the excess symbiotic organism would be released into the environment also. That's why I told it would be occurred. The symbionts not only providing the nutritional requirement that I would be telling later, but the fitness of the host, that is very very important, because we would be struggling with the insecticide resistance or host plant resistance, so many types of resistance, resurgence and other things. There, the microorganisms, they would be playing a very important role. A stellar role is being played. It increases the fecundity, it increases the longevity, it induces the female biased sex ratio. Then it encourages the mating frequency also, and a greater immunity it provides. And in the ecological sense, it increases the plasticity, so the host range would be widened. So all this because of the symbiont we have seen in the Paracoccus marginatus. What is it? Yes, we had recorded 136 host plants, sir, in that very brief period. It would be much more. And we came to know why, because of the insect feeds on numerous host plants. And the same way we did the metagenomic analysis of the Bemisia tabaci also, it is having more than 900 host plants, host. So this is in case of a predatory mite, the Metacillus occidentalis, the cytophaga. It is like a bacteria. I mean, it's a bacterium. It is called CLO, cytoplasma, cytophaga-like organism. When it is present, the oviposition would be increased. You see, the I means infected. U means uninfected. When the female is infected, the fecundity would be more compared to the uninfected females, and both the uninfected female and mated with the infected male. And the longevity also, when Wolbachia is present, the longevity is increasing in Aedes. The female biased sex ratio, I was telling, it is in the red period. The presence of Wolbachia, it results more of female. Then, in case of Drosophila, the Wolbachia increases the frequency of mating. Then the immunity to the host, because most of the insects, they act as a vector, but they are not getting infected, right, with the virus or the bacteria, whatever it is carrying. So it is because of the symbionts which provides the immunity to the host. See, in the case of uh, uh, the Aedes, the bacteria, the virus, Plasmodium, the filarial nematodes, they would not be affecting the host. They would just transmitting the pathogens only. The specific role, if you take in insects, it provides nutrition, it provides a protection against the natural enemies, it suppresses or manipulates the insect plant interaction. And most important, as of plant protection people like us, it is very useful for pesticide degradation in the environment. And one study in UP, where the organochlorines were dumped, nothing was there. And in that dumping yard, they using this technique, the microorganisms were recovered, and they were using this pesticide as a carbon source for their survival. The few examples what the microorganisms or the symbionts they are providing to the host. The in this easy fly, it is a blood sucker, and naturally the vitamin B would be deficient, isn't it? Is it? Our blood is deficient B. Yes, it is. That's why we are taking the B complex. It's a water soluble. Excess would be going out. Whatever little we required, it would be taken by the body and it never be in the blood. So the ZZ fly, it depends on Wigglesworthia glossidia for the B complex production. The same way, the mealybug, they would be relying on the microorganism for amino acid synthesis. This is a very interesting thing. So, how the microorganism protects the host? Here, the Oryzophilus surinamensis, the sawtooth head beetle, if the microorganism, the symbiont candidatus, if it is present, the sclerotization would be faster. So the Bavaria or the spiders, they will be finding very difficult to decimate the beetle. Whereas if the candidate is not there, the uh, cuticle formation would be delayed and it would be easy. That you would be knowing. So this is a very classical example how the symbionts would be helping against the natural enemies. So this is again how they escape from the pathogen, the vectors, how they are escaping. So they will be producing the reactive oxygen species, and it in turn it triggers the toll pathway, and it gives out some of the compounds like defense in cyclopin, and it inhibits the dengue and Zika virus, which affects the tissues in the host. And this is another example how the microorganism or the symbionts, they would be helpful to protect the host. So here the PAFID, it is being parasitized by Aphidus airvi. And if the symbiont Hamiltonella diffensa, if it is there, along with the APSC, it is a Acetosyphum pisum secondary endosymbiont. If these two symbionts are there, then the parasitoid will not, cannot develop on the P aphid. So this is again on nematode in Drosophila, and if the nematode, it would be developing in the host, but if the symbiont spiroplasma, if it is there, it cannot. And this is how the pesticide degradation happens. So most of the uh, symbionts, they would be utilizing the pesticide as the source, and they would be converting. So the pesticide would become your nutrition like. So many examples. I'll not go in detail in the host plant interaction, how they are suppressing the J and SA defensive pathway. So presence of symbionts, they would be suppressing this volatile, and they would be suppressing the defensive pathway, and the insect survives. And the symbionts are present in a variety of insects. It is not only crop pests, it is present in the beneficial insects, and it would be presenting on the some other insects which grows the fungus also. So many examples are there, and the pathway also would be different. So not only in the crop pest, in the beneficial insects also. In the beneficial insects, especially the honeybees, they would be very useful for surviving against some of the dreaded uh, the diseases like the American fall brood and European fall brood, that you'd be knowing that if AFB comes, then we have to just destroy the colony, except that nothing would work. So many insects, many phloem feeders, in the grain feeders, the amino acids and other things would be provided by the symbionts. And in I'll come to this later. A very good example has been given. So in case of honeybees, the antimicrobial compounds are being produced. So this is how some of the honeybees, especially our Indian honeybee, they would be escaping from the AFB. And in case of the bumblebees, they would be forming a biofilm. Biofilm, that also I'll come later, because of the presence of symbionts in the gut. So this is a classical example how the infection by the fungus is thwarted by the presence of symbionts. The leaf cutting ants, they would be growing the fungus. No, it is a sort of conversion of the cellulose, and it is threatened by a fungus called Escopis. So this Escopis, it would be inhibited by the presence of this compound, dendromycin. It is produced by the actinobacteria present in the ant, the pseudonocardia. So when this bacteria is present, it would be escaping from the fungal infection. In case of termite also, the termites, they'd be growing in the fungus, termatomyces. The termatomyces would be threatened by the presence of trichoderma, and it is it saves the microbes, saves the fungus colony in termites nest. How we can utilize the the study or the knowledge of symbionts in pest control? Whether we can use it? Definitely we can use it. Using some heterologous organisms, use of antibiotic, and the CRISPR-Cas, the RNA interference, like that we can use a number of techniques to utilize the symbionts in pest control. So in case of heterologous microorganism, here the microorganisms would be trans-infected. the presence of microorganism from one organism would be transferred to another microorganism. For example, in BPH, we know it is very important, not only the primary damage on the rice, it also transmits the viral diseases. Whereas the small brown plant topper, it harbors a Wolbachia. It carries a gene which can be infected in the Nilapavada lugens. So it cannot transmit the virus. So the virus transmission would be stopped there. The antibiotic, definitely, if we know clearly and correctly the microorganism present in the host, and we can use the specific antibiotic to target that particular microorganism, these symbionts would be killed, so the organism would not survive. Then CRISPR. So this is a very important here. It is a gene editing technology that you would be knowing, isn't it? So we manipulate the gene so that it would not form the biofilm. So this is a very important thing. So the here, the EDS, it requires a biofilm in its intestine, and if we the CRISPR using CRISPR, we can edit the gene so that the poor biofilm would be formed, and that would be detrimental for the EDS. And RNA interference, this is a very important technology, and we direct the RNA so that the eukaryotic organism would be affected. The varroa mite is a very important thing, and it would be the RNAi technology would be used to interfere with the varroa mite on the honeybees. So apart from that, so many other things are also there, using the Wolbachia as a viral inhibitor, and it will be used as a IIT, incompatible insect technique, and virus also we can use for the management of certain pests, and we can use numerous available parasitic symbionts also. There is another technology called paratransgenesis that also used for the management of insect pests. So this is how the Wolbachia would be used. The Wolbachia from the Drosophila would be acquired, and it is transferred into the mosquito, and the mosquito male and female would be having the Wolbachia, and the females would be retained. It would be tens of thousands of generation of mosquito would be bred, and only the males would be released, and the males would be having the incompatibility, and it would be competing with the wild population, and it was very successful in many of the countries, and for bird control, this is one of the very successful management aspect for the mosquito. And in the case of polyadenovirus, so here it is a very interesting thing. The wasp would be carrying the virus, and it would be aggregating in the calyces of the female wasp, and when it parasitizes the Lepidopteran caterpillar, it would be transferring the virions also, and the virions would be helpful for the compatibility of the egg or the larva developing inside the larva, and subsequently developing into the adult. So this is how the polyadenovirus would be transferred, and the adult wasp would be emerging from the Lepidopteran caterpillar. So this is a list of parasitic endosymbionts, symbionts. This we would be knowing, isn't it? The BT, from BT to the nematode, EPN, they are all parasitic. And in case of paratransgenesis, it is the protein we identify. The protein is important for the successful transmission of pathogen by the vector. So that expression of the protein would be interfere with that, and we are managing the number of the vectors like the rodnius and the Anopheles. This benefits, it costs. It costs the insect. It is not a free. So whatever the resources they would be using for the survival, growth, and the reproduction, a part of the resources would be utilized for the protection by these symbionts. These symbionts definitely they draw the energy from the insects, but when you compare to the death and the survival, definitely it benefits the host. In our laboratory, we work on many organisms. We started with the Helicobacter, and we worked on many insects, and we have now working on the mammals and birds also. So it has a lot of scope. The scope is unlimited for this type of studies. And this is our team. Many of my PhD students, and they worked on and they are working on this aspect. And a few publications, I have not listed all publications, so definitely it will be accepted. You try to work on that. So if you find anything interesting, you dig out the microorganism responsible for that. Previously, I used to tell you have to dig out the physiology of whatever you are doing. Now I'm telling you just see the microorganism responsible for that. So there is a scope. The traditional entomology, we have to just live, and we have to get many things from other disciplines also. And microbiology is a very, very important aspect that you have to do for a few things in your work, research work. So, to conclude, the symbionts are recognized for their role in insects. Thus, targeting symbionts could be a viable technology in the pest control, and we can use it as a defensive symbionts in the other insects, in the parasitoid predators and the productive insects. So it is a very good thing, and many labs are working in IRA. Definitely there is a lab works on the endosymbiont. So thank you very much for giving me the opportunity. Hope you have few queries to clarify also. All my seniors, contemporaries worked on different aspects, and the students also. No? So you would be having many things to be clarified. Ever worked on microorganism? Anybody in this group? No. You working on what aspect? Viral transmission. Definitely, it is a very good.
So through metagenomics, we can identify a lot of group of bacteria. Sir. How can we identify their functional roles? Because many of them are non-culturable. How can we?
So there is a database where all microbes, whatever the microorganisms so far identified, and their probable roles, so that you can pick up few things from that. The functional roles, to define a functional role for a microorganism, it would be very difficult. And whatever available literature, available database, from that you can get it. And you can also propose, suppose you have some microorganism, it's a dominant, or it is showing particularly to that organism, then you can relate it, and you can dig out the literature also. In that way you can fix the functional role for that particular microorganism. They would be having multiple roles. It is not a specific role. For the example, in case of Wolbachia, I have used Wolbachia in almost all slides. So it is having numerous functions. So like that.
So one more thing,
Yeah.
sir. Mostly for virus transmission studies, only that 7 endosymbionts are remotely studied, sir, like Rickettsia, Wolbachia, Apothecia, like that, sir. Some article they mentioned these symbionts will increase the transmission. In some articles they mentioned these symbionts reduce the fitness of the whitefly. So can you tell about this?
So definitely it will be increasing the fitness of the host. We had seen the searching ability of the white fly for on host. So we kept the cassava sets different resistant level, and we released the normal white fly and the white fly carrying the virus also, the infected one. The infected one, it was having a behavior which results in numerous feeding punctures. So that means it becomes a zombie. The virus, it dictates the vector to spread more. So like that, it increases the feeding punctures. So more feeding puncture, more the disease transmission. So many behavioral changes happen when it gets infected, apart from giving the fitness for the host.
Thank you, sir.
Mani, good afternoon. It was an excellent lecture. So my question would be, so we, based on our work, we are able to understand that a particular endosymbiont is giving adaptive advantage to the pest under means our target pest. So what are the ways to clear off that, let's say, symbiote out of it? So is it only molecular or is there any other way?
So if it is in the accessible area, we can go for the antibiotic. Antibiotic is one of the way we sterilize the organism, so that we can get the desired result. That it happened in a few examples. So we used the some of the antibiotics like. So that is one thing we can do, and
the molecular methods like the dsRNA techniques also can be used.
yes. So for short-term success, we can go for the antibiotic treatment.
So is there an example of using dsRNA and it being stable in the environment? Like
Whatever we used, the RNAi technology, it was relatively stable.
Symbionts are maybe it increases the fitness and it increases the replication of viruses. But in some cases, in some articles, they have mentioned that like Bacillus species, they are using for control of virus also. Can you elaborate this? Whether it is responsible for replication of virus in insects or they are used for control?
We have seen the symbiosis in three different things, no? Sometimes it would be antagonistic also, where the symbiont would be getting benefited, not the host. So everywhere it will not be mutually benefited like. So in that case, it would become the other way around.
Subgroups of Wolbachia, how do you relate to the diversity of insects? Here mainly we concentrate on the endosymbiosis in whiteflies or in disease transmission, etc.
Mosquitoes.
Mostly. But the diversity of Wolbachia and the insect diversity.
Oh, that I have not looked into. There'd be definitely there are a lot of subgroups, and what their role in this behavior. No, I don't have idea.
Okay. So because now just based on one primer amplifications, so they're not accepting that Wolbachia is a major group in most of the insects. So almost they have seen different types.
Yes,
Okay, maybe more than 10 types. So that
definitely.
they are
Yeah.
seeing that how
Yeah.
many types are present in each and every host.
Because
So
it is
that-
one of the most ubiquitous symbiont. It is present in almost all insects, and it has a diverse functions also. So definitely, as rightly told by you, there'd be a lot of subgroups. Hi, Vignesh.
Sir, good afternoon, sir.
Good afternoon.
Sir, I am from Asian Darai, Madurai, sir.
Tell me.
Sir, my doubt is whether the gut endosymbionts in all caste of honeybee same or it may be different, sir.
See, in this study, it is just like you cannot take the same water from the same well again. So every time it gets changed. So it is a sort of entropy in the nature. So each individual would be having a different type of the symbiont makeup. So we cannot generalize the same, but we can tell this group is present like that. So every individual will be having a different type of the symbiotic makeup.
Okay, sir. Okay,
Am
sir.
I
Thank you, sir.
clear? Yeah. At least they should fix up a general function of that group. It's a very, very, yeah, very laborious. If we get the microorganism, we tried, and the last of my student, she tried. It's a really laborious process.
And costly also.
Yeah, the
It's very
medium,
costly.
yeah, media also we have to select.
You cannot mimic the insect system.
Yes, exactly.
That's very difficult. Because I tried it in 1986. Probably in this group, I am the oldest to work on endosymbionts. Yeah, I thank my professor, Dr. Jairaj, who is in the photograph, and many of you may not be knowing, students. And he gave me the subject of endosymbionts. When I took the subject of neem to work on BPH, brown plant hopper, one fine day he called me, and now from today onwards, you are going to work on the effect of neem on endosymbionts. The whole of the thesis will be converted into endosymbiosis and neem. So I was little baffled because my title of MSc thesis was, during those days MSc is equivalent to 1.5 times of PhD. So that much work they give our masters. They are very taskmasters. That my title was changed 3 times. Really, I enjoy those days. At the time, really, it was very tough. Now, when I recall, I had been exposed to different types of research areas. Neem was given one title, Neem versus pest, Neem versus nephrocrosis, Neem versus Marasmia patinalis, Neem versus BPH, Nilparuda allugens, Neem versus GLH, nephrotics virulence. Then suddenly he said that, okay, this and all you have done, these are all very normal procedure. Testing insecticide, like insecticide you are testing, you are going to test neem with pest. We should try something which is very new. Those who are not done first, you should give it to the world, a very first new kind of research. Then I recollect one day that it was around 10:30 in the night. Myself, S.V. Krishnamurthy, and Dr. Manikam, was that Rajamanikam? We three are sitting and looking at the microscope, counting the number of symbionts affected by neem. So I used neem extract to kill the symbionts, to create a symbiotic BPH. That was not possible, but there is some reduction was observed. We are counting. 2 days before he told that I will check your number because this is first of its kind report, world record. Then he said that I will not leave you and you should not give me any bogus data. I will come and check you whether you are exclusively counting through microscope. Then to my surprise, he came around at 10:30 in the night, our old entomology, UG, present day UG laboratory. Those days we used to sit there. By 10:30 he came and he verified by looking into the binocular and looking into the East Lake symbionts, wireless. The East Lake symbionts were the harbored by brown plant hopper. So that was the task day. I take this advantage of the platform to recollect my days. Thanks. When I asked Dr. Mani to give a lecture, he gave the title Endosymbionts, the hidden players. I was um, really interested to see how you have developed metagenomics from 86 to this day. At the time also, he asked me about metagenomics. I didn't know the meaning of that time. What the metagenomics and all. I simply said that there are so many work. Now I can't complete this thing. Now you have done an excellent work and given to so many students. Thank you, Dr. Mani, for having a excellent lecture.
Mosquito, is there any other practically using the endosymbionts for the management of insects? In IIT technique you told about.
That is the one thing. It has been commercialized. Other things, no, not it. And it was from the Western Sydney University. The Regler, he was the person behind the Wolbachia induced the incompatibility. And no other things. That Chagas disease they were trying in Rodney's and ZZ fly also. So all the public health related work only, not in agriculture so far. And they wide scale and for important pace. Definitely in the public work, public health, it is working. Yeah, they were in the resistance. They were trying because it is one of the most notorious pest, overcoming all insecticides. Ma'am, the regarding the vertebrate pest management, that I'll brief you later. So if I start, then it will be endless because it would be.
Good afternoon to everyone. It's time of vote of thanks, formal vote of thanks. Thanks for, first of all, I thank Professor Andet for giving me the opportunity to say vote of thanks to my good friend, Mani Chalapan, my PhD classmate at IRI. We spend a lot of time and energy there at IRI. So if I recollect that, it will be a very big story. I I just cherish my memories with him. Those days were very enjoyable at IRI, New Delhi, for our PhD program. So Dr. Mani has done an extremely very big travel in the field of endosymbionts. Nobody had done so far this much travel in endosymbionts because he has listed around 15-20 students. If he has done an excellent work with those students, really it would find a very big voluminous work of endosymbiotic work in India. There is very few entomologists they are working on endosymbionts. Many microbiologists are working with that, but the combination of microbiologists and entomologists is really lacking because we do not know much of the microbiological side and they do not know much of the entomological side. Unless otherwise there is a balance between these two persons, this subject cannot develop well. But this is a very extremely good field that everyone should think of. At least I request director and head of the department a portion of subject that can be focused, not that full-fledged thesis. A portion of thesis can be focused to understand the role of endosymbionts. Okay. I thank Dr. Mani has come all the way from Thrissur today morning. He started his journey by 6 o'clock and all the way he traveled and arrived here by 9, and he has to again go back. So he is very, very good traveler, and everywhere he goes by his own car or his own personal mobility, and extensive traveler. That is why he says that I should not transmit disease to other putting mask. When I asked that, are you protecting yourself? No, no, I am protecting others. So by the way, I thank Dr. Mani for having taken care of others well. I thank our director and professor and head and PG coordinator and other members and other teachers, students, those who have taken your time and be present for almost around 2 hours or 2.5 hours here since the start of our final OC with Sanira, and in continuation to the program and endosymbionts presentation by Dr. Mani. And I thank each and everyone for having taken your time to be with us. Thanks one and all.